The next thing I'm going to talk about is synaptic activity, also called synaptic transmission. Basically, it's what happens after we've gone through an action potential. So the electrophysiology of neurons involves the resting potential, the local potentials, and then the action potentials. And then this is followed by synaptic transmission. So that's what we'll take a look at now. Synaptic transmission is what occurs at the very ends of the axons. So the action potentials exert their effect at the ends of the axons where we have a synapse. A synapse is a meeting point between the end of one neuron and either another neuron or a muscle cell or a gland cell. So basically, a synapse is a place where a neuron communicates with another cell. An example of a synapse we already know is a neuromuscular junction. That's where a neuron meets a skeletal muscle cell. There are two main classifications of synapses in the human body: chemical synapses and electrical synapses. Chemical synapses include the neuromuscular junction that we've already talked about in this class. Chemical synapses involve the use of a neurotransmitter crossing a synaptic cleft. Electrical synapses occur when one neuron directly touches another neuron and the electricity just travels from one neuron to another. These are really rare though, so we won't even talk about them. We'll spend most of our time talking about the chemical synapses. So in terms of their structure, a chemical synapse has three main parts: the presynaptic neuron, the synaptic cleft, and then the postsynaptic cell. The job of the presynaptic neuron is to release neurotransmitter. The neurotransmitter then travels across the synaptic cleft, and then it goes and it binds to receptors on the postsynaptic cell. So the postsynaptic cell has receptors that bind to neurotransmitter. The postsynaptic cell may or may not be a neuron. If the postsynaptic cell is another neuron, an action potential may or may not be triggered in that other neuron. From this point forward, we are going to assume that the postsynaptic cell is a neuron and not a muscle and not a gland, just for simplicity's sake. So what exactly do these neurotransmitters do? Right? I like to say that the neurotransmitters do nothing other than bind to a receptor. I know that sounds kind of strange, but I like to emphasize the fact that it's the receptors that have the effect on the postsynaptic neuron. Right? So the job of the neurotransmitter is just to activate the receptor. And if the receptor opens sodium channels, that will cause depolarization in our neuron, and that will be excitatory in nature. But if the receptor opens chloride ion channels or potassium ion channels, that will be hyperpolarizing in nature, and that will inhibit action potentials in our postsynaptic cell. So it's the receptors that are important here. So I want to talk about a type of synapse called a cholinergic synapse. A cholinergic synapse is a synapse that uses the neurotransmitter acetylcholine. The neuromuscular junction is a type of cholinergic synapse. Now it turns out that the receptors for acetylcholine, these cholinergic receptors, they can be excitatory or inhibitory. We are going to take a look at an excitatory cholinergic synapse. So we're going to look at a situation where the receptor is excitatory in nature. All right, the steps are here. Let's take a look at them in more detail here. So step number one of synaptic transmission at a cholinergic synapse is the action potential arrives at the knob. So in this picture we can see the action potential is arriving at our synaptic knob. That's the red in the picture. Step number two is that calcium comes into the synaptic knob. And the reason why calcium comes into the synaptic knob is because there are voltage-gated calcium channels on the cell membrane of the synaptic knob. Those voltage-gated calcium channels are opened by that action potential that has come down to the knob. So now we can see calcium is entering into our synaptic knob. The next thing that happens is that the calcium acts as an intracellular messenger inside the knob, and this messenger basically tells the knob to release the neurotransmitter. So the calcium triggers the exocytosis of the neurotransmitter acetylcholine. So we can see that the acetylcholine has been exocytosed into the synaptic cleft. In the next step, the acetylcholine will bind to receptors on the postsynaptic membrane, causing depolarization of the postsynaptic membrane. Now we're assuming that this is an excitatory cholinergic synapse. And for it to be excitatory, that means that the receptor has to open sodium channels. The sodium then rushes into the postsynaptic neuron, causing excitation and contributing to the likelihood of an action potential. It's not a guarantee that this other neuron will get an action potential, but it contributes to the likelihood that there will be one. And so those events are pretty typical at a synapse. So now I want to talk about stopping synaptic transmission. Basically, synaptic transmission stops when the presynaptic cell stops releasing neurotransmitters. And then we have to get rid of the neurotransmitters. Now, neurotransmitters only bind to the receptor for about 1 millisecond, and then they unbind, and now they're in the cleft again. And so what can happen from here is that the neurotransmitter can just diffuse away, or the neurotransmitter can be taken back up by the presynaptic neuron. And then the presynaptic neuron can actually reuse the neurotransmitter. Also, the neurotransmitter can be degraded by enzymes in the cleft. And I didn't really talk about that in this picture, but you can see it, the little yellow banana-looking like things. Those are enzymes that can break down the neurotransmitter. In this case, these particular enzymes are called acetylcholinesterase. And they break down acetylcholine into smaller pieces, and those smaller pieces are actually usually taken back up by the presynaptic neuron. So here we can see a synapse. This is our presynaptic neuron, this is our postsynaptic neuron, and here's our cleft. You can see our synaptic vesicles inside the presynaptic neuron. These are our voltage-gated calcium channels, okay? And these are the neurotransmitter receptors, in this case acetylcholine receptors. So what happens first is the action potential travels down the neuron and gets to the very end of the synaptic knob. That action potential opens our voltage-gated calcium channels. Calcium then comes into the synaptic knob, and the calcium triggers the synaptic vesicles to exocytose their contents into the synaptic cleft. So now the acetylcholine, shown in yellow and orange, is in the synaptic cleft. The acetylcholine then will go and bind to receptors on the postsynaptic neuron cell membrane. These acetylcholine receptors are also chemically gated sodium channels. So when the acetylcholine binds, the gates open and the sodium enters. And the sodium will depolarize this other neuron, leading to excitation.